Hello everyone and welcome. Today we are going to take up one of the most important concepts in all of biochemistry, and that is enzymes. Let's try and make this topic crystal clear so it becomes very simple and memorable for your exams.
Okay, so we will not start with a direct definition. Instead, let's begin with a small chemical puzzle, a mystery that we will solve together in this session.
Just imagine you have a solution of hydrogen peroxide. You know, on its own, it breaks down very, very slowly. But now, what if you add just a pinch of a special powder called catalase? The moment you add it, the solution starts bubbling furiously, releasing lots of oxygen gas. There is no fire, no high temperature. So how is this reaction happening so fast right at room temperature? Just think about it for a moment.
Now, this little puzzle brings us to a fundamental problem of life itself. You see, for you to be alive, for your cells to work, thousands of chemical reactions must happen every single second. But the condition is they have to happen at normal body temperature, not inside some boiling flask in a lab. So the big question is, how? This is the central problem that enzymes are here to solve.
So, let's reveal the solution to our puzzle. That special powder, the catalase, is actually an enzyme. And enzymes, well, you can think of them as secret tunnels that our body uses to make very difficult journeys happen very, very easily.
Now for the proper exam-safe definition. An enzyme is a biological catalyst. Let's break it down. Biological simply means it's made inside living cells, and it's usually a protein. And catalyst means its job is to speed up a reaction, and it can do this job again and again and again without getting used up itself.
To understand how they work, let's use a very simple picture. For any chemical reaction to even start, the molecules, the reactants, they need a certain minimum amount of energy. It's like they need to climb a huge mountain to get to the other side. This energy mountain has a name. It's called the activation energy.
So what does the enzyme do? Now this is important. It doesn't give the reactants a push to climb the mountain. No. Instead, it creates a completely new path, a tunnel, straight through the mountain. And this new path, this tunnel, has a much lower activation energy. So naturally, the journey from reactant to product becomes incredibly fast and easy.
And this picture shows us a very crucial point. The enzyme does not change what you start with or what you end with. The starting point and the ending point are the same. It only changes the path. This is a common point of confusion for many students, so please remember, enzymes do not provide energy. They just lower the energy barrier for a reaction that was going to happen anyway.
So we know that enzymes create a kind of tunnel, but how do they actually do this at a molecular level with such amazing precision? Let's build a clear mental image for this so you never forget it.
I want you to imagine that the inside of your cell is like a very busy restaurant kitchen. And in this kitchen, the enzymes are like highly specialized chefs. The important thing is that each chef knows exactly what to do, but only with one specific ingredient.
So let's look at how this chef works. It's a 4-step cycle. Step 1. The ingredient arrives. In chemistry, we call this the substrate. It fits perfectly into a specific pocket on the chef. This pocket is the enzyme's active site. Step 2. The chef prepares the dish. He holds the ingredient in the exact right position, maybe twisting it or stressing a chemical bond to help the reaction happen. This is the catalysis part. Step 3. The dish is served. The finished product is released from the active site. And finally, step 4, the chef is completely unchanged. He is free and ready for the next order, for the next substrate molecule to come along.
Now, what is truly remarkable here is the speed. This whole cycle, it doesn't happen once a minute or once a second. For a single enzyme molecule, this can happen hundreds or even thousands of times every second. This incredible efficiency is what makes life itself possible.
All right, we have seen how important these molecular chefs are, but what happens if a specific chef is missing from the kitchen? Let's look at a very common, real-world example that you might have seen.
Many of us know someone, or maybe it's you yourself, who finds it difficult to digest milk. After drinking milk, you might feel discomfort or bloating. Why does this happen? The answer, as you might have guessed, is a missing enzyme.
Let us look at this comparison here. Milk contains a sugar called lactose. Now, in most people, the body produces a very specific enzyme, a chef named lactase. Its one and only job is to break down lactose into simpler sugars, which our body can easily absorb. But in people with lactose intolerance, this chef, this lactase enzyme, is either missing or it's just not working properly. So what happens? The lactose ingredient is never processed. It just passes through the body undigested, and that causes all the problems. So you see, this is a perfect illustration of enzyme specificity. One chef, one job. If that chef is missing, the entire production line for that one dish comes to a stop. Luckily for us, modern technology now allows us to simply take a lactase supplement. It's like we can swallow the missing chef, you could say, to help us digest milk.
Okay, so we have covered a lot of ground. Let's now bring it all together and quickly recap the most important points that you must remember, both for your exams and for your general understanding.
So please try to remember these three things. First, the core idea. Enzymes are simply biological catalysts that speed up reactions by creating a low-energy tunnel through that activation energy mountain. Second, the mental picture. Always think of that highly specific chef in a busy kitchen, perfectly preparing just one type of ingredient. And third, the real example. Lactose intolerance is caused by a single missing chef, the enzyme lactase. I can promise you, if you can clearly recall these three pillars, the main idea, one clear picture, and one solid example, this entire topic of enzymes will become much, much lighter for you, and you will feel far more confident when you are preparing for your exams. And to leave you with a final thought, we have seen that our life depends on thousands of these different enzyme chefs. Each one is a specific protein, folded into a very precise shape. So the final question is this: where does the cell keep the master cookbook? What do you think is the single most important master molecule that holds all the recipes, all the instructions, to build every single one of these enzymes correctly? Think about it. We will explore that in our next